So next we start with pteridophytes. So dear students, we have studied in detail about algae and bryophyte. Now we are talking about a third group of pteridophytes. Now when we talk about general characteristic, then we come into some points. So here we start with the first point, that is these are the most primitive vascular plants. Why they are most primitive vascular plants? Because these are the first vascular plants. Vascular plants means they have vascular tissue, which was not found in case of algae and bryophyte. This vascular tissue in case of this vascular plant are xylem and phloem. But in case of xylem, we remember that true vessels are absent. And when we talk about phloem, in phloem also companion cell and sieve tube are absent. Companion cell and sieve tube are absent. So in xylem, if true vessels are absent, means the tracheids are mainly playing a role in transport of water. When we are talking about phloem, in companion cell and sieve tube are absent, so this phloem of pteridophyte have albuminous cell and sieve cell in place of these companions and sieve tube, which are found in gymnosperm and angiosperm. Okay. Next, if we see, these are flowerless, means they are not able to produce a flower. Okay. Then with lack of flower, they are also a seedless. They do not form a seeds. They are spore producing. They are spore producing. They produce a spore. And they are spore producing land plant. But they are cryptogams. They are cryptogams. Cryptogams means seedless. And in this also, cryptos means conspicuous or clearly not visible, and gametes means particular visible structures are not there for the reproduction. Next we talk about then they are commonly called as botanical snakes or reptiles of plant kingdom, or vascular amphibians of plant kingdom. Okay. And so this is the particular other name of this particular plant. Why amphibians? Because still they need water for fertilization. They need water for fertilization. So due to this, they are a vascular amphibians, cryptogams. Their plant body is differentiated into true root, stem and leaf. Means they do not have a thallus structure. The plant body is differentiated, and term pteridophyte given by Haeckel. Term pteridophyte is given by Haeckel. Now pteridophytes require cool, damp and shady places to grow. They require cool, damp and shady places to grow. And they need water for fertilization as well. So because of this restricted requirement and need of water for fertilization, this pteridophyte spread of this pteridophyte is limited and restricted to narrow geographical region. Okay, so these are the things due to which they are found in a few areas only. Now they have true root, stem and leaf. So when we talk about a leaf, they have two type of leaves, that is, first classification, they have two type of leaves, that is trophophils and sporophils. So trophophils are the non-reproductive leaves, and these non-reproductive leaves, they do not play a role in reproduction. So the function of these leaves is a photosynthesis. But they have another type of leaves that are known as sporophils. Sporophils are the leaves which bear sporangia. Means they are a reproductive leaves. So they play a role in a reproduction. Okay. Now this is the classification, first classification of the leaf. Now second classification of leaf that we perform, that is with respect to the size of the leaf. So based on size of leaf are divided into two types, that is microphylls and megaphylls. Now these megaphylls are also known as macrophylls. Now these microphylls are smaller in size than stem. Compared to the stem, they are smaller in size. And if we see the example of this microphylls, then that are Selaginella, there are Selaginella, Lycopodium and Equisetum. So all these plants have small leaves, microphylls. So these all are termed as microphylous pteridophyte. Whereas megaphylls are the large in size than stem. So they are known as macrophylls or megaphylls. They are also known as fronds. Be remember, they are also named as fronds. So these megaphylls are found in fern plants. in fern plants, and fern plants like Dryopteris, Marsilia, Salvinia, Adiantum, all these fern plants have a particularly this megaphase, and that we will study in case of the last order, that is a Pteropsida. Okay, so two type of leaves based on the size, that is microphylous, and the example are considered as a microphylous pteridophyte, and in megaphase the examples of fern plants are considered as a megaphylous pteridophyte. This pteridophyte are considered as a successful terrestrial plant, but they are not considered as, they are not completely successful terrestrial plant, because they need water for fertilization, and the time period till which in evolution they need a water, then they are not completely considered as a successful terrestrial plant. Okay, next when we talk about pteridophyte, then they have two particular bodies, that is sporophyte and gametophyte. Now when we compare a sporophyte and gametophyte of pteridophyte, it has come in consideration that sporophyte is obvious diploid and spore producing body, that we know. In gametophyte also it is haploid and gamete producing body, that we all know. But the important is in case of pteridophyte, sporophyte is dominant. In algae, in bryophyte, we have considered this gametophyte as dominant, but from this pteridophyte, the dominant phase is nothing but which one? Sporophyte. So means this sporophyte is independent, free living. If similarly, if we consider gametophyte, then gametophyte is a recessive, obvious, that is not a dominant, but this is also independent. This is also independent and free living. Means here both gametophyte and sporophyte are independent of each other. Next, if we see, both are able to perform photosynthesis, means both are independent, means they can perform photosynthesis, and this also perform photosynthesis. So one important thing which we derive from this part, that point that we should understand, that is both sporophyte and gametophyte are independent of each other in pteridophytes. So this is the most important statement with respect to question, and please remember this. Okay, so this is the about the particular sporophyte and gametophyte. Now if we know their general characteristic, their gametophytic nature, the sporophytic nature, then we are able to now draw their life cycle. So now next we are going to talk about what? A life cycle of a pteridophyte. So now students, we are starting with a life cycle of pteridophyte. So when we talk about life cycle of pteridophyte, first we know that they have a diploid sporophyte which is a dominant phase, and the gametophyte is also independent, but it is a recessive phase. So when we talk about this sporophyte, here you can see the sporophyte is diploid, and this is the sporophytic diagram, which have a leaves, okay, stem and the root structures, and this are root stock. These are root stock, that is an underground stem which is unbranched, then it is known as root stock. They have a coiling of young leaves like the spring of watch, and that is known as cercinatices. So you see here cercinatices as well, as that is a coiling of young leaves like the spring of a watch. Then when we see these leaves, as we have discussed already, that there is a reproductive leaves are known as sporophylls. Now here you can see these sporophylls are a part of sporophyte, so they are obviously diploid, And on a sporophyll, you will find a sum of group of sporangia. Sporangia is actually plural of sporangium. Okay, here you can see this is a single sporangium, and as plural of sporangium is sporangia. So when so many sporangia are found in a group, it is known as sorus. And the plural of sorus is sori. So you can see here so many sori are there. Now this sporophyll bear a sorus, that and the structure of sorus is like this. In this structure you can see here that is a stalk, and this structures, these are the known as sporangium. And sporangium as what? Sporangium are the nothing structure which are going to produce a spore. So if we talk about this structure again, this sporangium are protected by a one flap like covering that is known as true indusium. So if this is a part of sporus, then true indusium ploidy is also diploid. Now in this sporangium you can see this one, that are the spores. So we know that sporophyte has a spore mother cell and which undergo meiosis and produce these spores. So here earlier there was a spore mother cell which was undergone a meiosis and produced a spore. Now sporangium structure is like this. This sporangium structure is like this. They have an annulus, they have a cell, and inside a spore which are produced by the meiosis of spore mother cell. Now these stomium cell are somewhat elongated cells. When there is a breakdown of stomium cell occur, this annulus become free, and when annulus get out from this get removed from this particular plant and become curved, this spore which are found here that start flowing, and they blow from here and the spore reach to the suitable substratum and germinate, and after germination they form a prothallus. What they form? Prothallus. And prothallus is a nothing but a gametophyte. It is nothing but a what? Gametophyte. Now this gametophyte structure, when we study, in this gametophytic structure you can see they have a prothallus, that gametophytic structure, and this prothallus have a rhizoids. They have a rhizoids, and in between this rhizoid you will find a male sex organ, that is antheridium. But on the anterior part of this prothallus you will find a archegonium, whereas in the posterior part you will find a antheridium. Now we know that archegonium is a female sex organ, so this female sex organ produces an egg. But archegonium is haploid, so egg is also haploid, so it is produced by the mitosis. But when we talk about antheridium, antheridium is haploid and it forms an anthrozoid mother cell, anthrozoids from the anthrozoid mother cell by mitosis. These anthrozoids are a male gamete and they are multiflagellated. Now this multiflagellated anthrozoid, when fuse or when fertilize or syngamy with the egg, they produce a zygote. This zygote undergo mitosis, produce embryo, and embryo will develop in a sporophyte. So again sporophyte will be obtained. So this is the way how the whole life cycle of pteridophyte goes. Now what the questions which are being asked from this? So first is the sporophytic generation is here, gametophytic generation is here, dominant is which one? Sporophyte. So diploid phase is dominant. So commonly according to NCERT, this life cycle is known as haplodiploidic, but actually we can call it as a diplohaplodic. Okay. Next, what is the ploidy of prothallus? That is haploid. What is the ploidy of this sporangia, indusium, stomium cell? That all they are, so that is what? A diploid. But this antheridium and this all particular archegonium are haploid. Now the important point that you should remember, that here male gamete or female gamete, anything that is produced by mitosis, not meiosis. So in pteridophyte, as well as we have studied in case of bryophytes, also gametes are produced by mitosis, not by meiosis. Here what is produced by meiosis? Spore are produced by meiosis. So their life cycle can be considered as a sporic meiosis. What we can call is a sporic meiosis. So this is the complete picture of this particular life cycle. You can go through this and you can study it. Okay, so now this life cycle is over. Now we skip or we write some of the inferences from this life cycle, and then we move to the classification. Now we note down some of the inferences from the life cycle which we have studied about pteridophyte. So by studying life cycle, we get to know that the life cycle of pteridophyte is haplodiploidic according to NCERT, but it can also be called as diplohaplodic, as is main phase or dominant phase is a diploid, that is a sporophyte. Okay, Now next, after to that, we come to know that prothallus is a gametophyte, and this prothallus, it is a gametophyte which is a haploid. It is independent, green, multicellular and unconspicuous. Now this have a non-vascular gametophyte. We remember vascular tissues are present, but vascular tissues are present in case of which one? In case of sporophytic part. Their gametophyte is non-vascular. They also show succinate-tices, which we have marked in case of sporophytic diagram. This succinate-tices is a what? Coiling of young leaves. Coiling of young leaves. And this coiling of young leaves protect the tip of a tip of shoot from mechanical injury. Okay, now in this one more structure is found that is known as ramenta, and ramenta are the multicellular, they are multicellular brown hairs. It are brown colored hairs which are present on stem, root, leaf, petiole, etc. And it also prevent from mechanical injury. They also protect or prevent from mechanical injury. So these are the points which we have found in this one. Now, in case of pteridophyte also we are saying that fertilization is occurring but water is needed, means in this also they have a zoidogamy, that is a need of water for fertilization of male and female gamete. And during this zoidogamy we found that this multiflagellated anthrozoid or male gamete reach to neck of archegonia by chemotactic method. This chemotactic method we have studied in case of bryophyte as well, where anthrozoid move to the neck of archegonia by a chemotactic method. And in this chemotactic method, where chemical stimulus is malic acid. Now in place of this malic acid in bryophyte we have studied a potassium and sugar. Now here there is a malic acid. Okay, so now this is what we have understood from the life cycle of pteridophyte. So till now characters are over, life cycle over. Now we are coming to the last part, that is the classification of pteridophyte. What we are going to study now? Classification of pteridophyte. Now this Pteridophyta is classified into 4 classes. That is the first class is PSILPSIDA. Second class is LYCOPSIDA. Third is Spinopsida, and fourth is Pteropsida. So now we are going to talk about all these four classes one by one, and we are starting with first, that is Psilopsida, then we talk about Lycopsida, then we will talk about Spinoffsida, and at last we will talk about Pteropsida. We remember last Pteropsida is a class of fern pteridophytes. Which one? Ferns. So now if we start with the first group, that is Psilopsida, then this Psilopsida, in this majority of them are extinct. This is the extinct group. Majority of them are extinct. They are not living. And example, if we see in case of Psilopsida, which is the most ancient group, first is Xylotum, that you remember, because this is an important plant, as this is the living fossil. Out of this Xylotum, there is a Rhynia. One more pteridophyte in Psilopsida is a Rhynia, and next is Horneophyton. This Rhynia and Horneophyton are fossil plant. So there is no need to remember this two as much, but this is much important. They can ask from this questions. Okay, so this is the first group, that is Psilopsida, and in this living fossil is a Xylotum. Now we come to the next group, and when we talk about the next group or a next class, so next class is Lycopsida. Now in this Lycopsida include club mosses. Which mosses? Club mosses. So whatever the example which we are going to study in this class, they are club mosses. And in this club mosses, if we take an example, then first example is a Lycopodium, from which the term Lycopsida arises. Now Lycopodium is considered as a commonly observed club moss, so it is known as common club moss. Okay, with this, another example is Selaginella. Another example is a Selaginella, which is the one of the important example of this. Third, we can write about Isoetes. Now in Isoetes, we remember that it is also known as Marlin grass or quillwort, and this is the only pteridophyte, or it is one of the exceptional pteridophyte which is found in Lycopsida, which show secondary growth, means that can form a thick woody stems. Okay, so secondary growth is found in case of Isoetes. So now out of these three, for our study purpose, for NEET purpose, this Selaginella is much important, and so many questions are being asked from the Selaginella. So it is our duty to study this Selaginella in detail. Okay, so we will study Selaginella somewhat in detail with respect to some important point and some important aspects. So Selaginella. Now this Selaginella is also known as, spike moss. What we call it? Spike moss. Okay, it is also a club moss, but it is specifically called as a spike moss. This Selaginella is a heterosporous pteridophyte. It is a heterosporous pteridophyte. Okay, now when we draw the diagram of Selaginella, we found somewhat this type of diagram in case of Selaginella. And in this diagram you will find a leaf that are small leaves, microphyls all over the body part. So this type of particular Selaginella is found, and the branching where you will find in Selaginella, from this one structure protrude downward, and that structure develop some of the branching at the bottom for the anchorage. Okay, now in this, if we see, this Pteridophyta have leaves, and these leaves are the small leaves, so it is known as microphylls, and this Selaginella is a microphylous pteridophyte. Now when you see, you will see here a group of sporophyll, and this group of sporophyll is known as strobilus or cone. What we call strobilus or cone, and strobilus or cone is nothing but it is a group of sporophyll, and we have studied that sporophyll are the leaves over which sporangia are present. Now this strobilus, in case of Selaginella, it is loose and small. Next to that, this downwardly protruding structure, it is known as rhizophore. And this rhizophore is also known as organ sui generis. Now this organ sui generis, it is known as organ sui generis because it has a character of both stem and root. Because of this, it is known as organ sui generis. Now this is the overall structure of this Selaginella, but now the leaves are of importance. So when we see the leaves of this Selaginella, in leaves you will find some small leaf-like structure at the base of this lamina, and this is known as ligule. What it is known as? Ligule. So what do you mean by ligule? So ligule are nothing but a small leaf-like structure present at base of lamina, okay. And when we see the structure of this ligule, in this structure we find somewhat structure like this, and the base of this ligule is covered by a structure that is known as glossopodium. What we call? Glossopodium. So glossopodium is a structure which covers the base of this ligule in a sheath form. Okay. Next, this Selaginella or Heterosporous pteridophyte. Okay. And in this, one species of Selaginella is there which is known as Selaginella bryopteris. This Selaginella bryopteris is the species which is also known as Sanjeevani booty, but it has no medicinal property. Okay. It is known that Heterospory of Selaginella and Salvinia is responsible for seed habit, and seed habit means formation of seed in the future, this particular gymnosperm and angiosperm. Seed habit is a precursor event for development of seed in gymnosperm and angiosperm. Means this precursor event, seed habit, a heterospory are responsible for evolution of a seed formation in case of gymnosperm and angiosperm. Now what actually happens in case of this seed habit? In seed habit, specifically in case of heterosporous pteridophyte, female gametophyte is retained over parent sporophyte. It is retained over parent sporophyte. And when the female gametophyte is retained over parent sporophyte, the seed was found, as in gymnosperm and angiosperm. In gymnosperm and angiosperm, if we see, female gametophyte is dependent on the sporophyte, and we can think that is the reason why they have seeds. A similar type of event was somewhat developed in heterosporous pteridophyte. So because of this, it is responsible for evolution of seed formation in gymnosperm and angiosperm like higher plants. Okay, so this is with respect to our second part, that is the Lycopsida. Now we come to the third class, that is Spinoffsida. Now in Spinoffsida, they include horsetails, and one of the important example of horsetail is equisetum. Now when we see the diagram of equisetum, we will find somewhat this type of diagram. In case of this equisetum. Now in this diagram, you can observe the presence of strobilus over the tip. And apart from this strobilus, they have some of the node-internode differentiation, and on node they have scaly leaves. Okay, they have some white of this type, scaly leaves. You can see this is a node, this is internode. So they have internode node differentiation, they have scaly leaves, they have strobilus, and when you see the underground part, then you will find a stem. That is, stem is named as rhizome. This stem is named as what? Rhizome. And this rhizome can have some of the adventitious branches. Now, if we talk about this equisetum, then in this equisetum, root, stem and scaly leaves are present. Which scaly leaves are found? Now next to that, node and internode are differentiated. Node and internode are differentiated. Then silica is present in cell wall of stem and leaf, and due to presence of silica in cell wall of stem and leaf epidermis cells, when wind blows, so due to wind this plant collide with each other, and due to collision of this plant with each other and presence of silica in this cell wall, they cause forest fire. What they cause? Forest fire. Okay, so that is the Spinoffsida or equisetum. Now we talk about a fourth class of this pteridophyte, that is Pteropsida. And as we have studied in Pteropsida, we talk about the fern plants. So they include fern plants. Now in these fern plants, they have megaphylls or large leaves. These megaphylls or large leaves are known as fronds. What they are called? They are called fronds. This Pteropsida are the plant which have ramenta, and we have studied ramenta are the brown hairs which provide protection from the mechanical injury. This fern plant obviously have circinate dices. And one more thing which we have studied, that is a root stock. Root stock is nothing but underground unbranched stem. If this underground stem is branched, then we call it as a rhizome, but it is what? Rootstock, which is a unbranched. Next to that, we have studied one important structure, that is a indusium, which protect the developing sporangium. So in this, indusium is present, and we have studied that indusium is diploid. Now this indusium are of two types. One is false indusium and one is true indusium. Now this false indusium, it does not develop separately. It is formed by the curling of lip margin. It does not develop separately, formed by a curling of leaf margin. So here we can take an example of the adiantum, and adiantum is known as walking fern. Next to that, indusium is of another type, that is a second type, and that second type is true indusium. This true indusium is found in case of Pteridophytes. It develops from the separate cells, so it is known as a true indusium. Next, we talk about some of the examples of Pteropsida. Any is this example we will discuss one by one all. First is Bryopteris. Second is Adiantum. Third is, now Adiantum is also known as walking fern and maiden hair fern. Third example is Pteris. then Pteridium, then Marsilia, then Azolla. Now, with the leaves of Azolla, a fern pteridophyte, Anabina azole, that is a blue-green algae, show symbiotic association for nitrogen fixation and conversion of this nitrogen into ammonia. Next to Azolla is Osmunda, and one more example, that is Ophioglossum. And Ophioglossum is a pteridophyte which has highest chromosome number, and this chromosome number are 1260 in diploid organism. Okay, so they have a highest number of chromosomes. So these are some of the example of Pteropsida. Now we are dealing a small last topic, that is a difference between Homosporous and Heterosporous pteridophyte. So Homosporous and Heterosporous, if we differentiate, then Homosporous pteridophyte produce only one type of spore, whereas Heterosporous pteridophyte produce two type of spore. So if there is a one spore, it will form a monoecious pteridophyte, means which have both male and female sex organ in the one plant, and these two type of spore, which are the microspore and megaspore, that will form a dioecious organism. And in dioecious organism, male and female sex organ are present on the two different plant bodies. Now if we take an example of this Homosporous pteridophyte, then that are Lycopodium, Equisetum, then Pteridium, Pteris, Dryopteris, Adiantum. These all are homospores. And when we talk about heterospores, the example important are Selaginella and Salvinia. Then Azolla is also there, Marsilia is there. All this comes under the heterosporous pteridophyte. But be remember that and fern are also and this particular thing. Now we remember one of the important statement, that most of the pteridophyte are heterosporous. Most of the pteridophyte are heterosporous. Okay, so that is about a pteridophyte, its classification and all the characteristics.